What is recombinant virology? Viruses are most commonly associated with disease, but imagine if we could use them to help us rather than hurt us. With recombinant virology, researchers work to understand viruses better so that we can predict disease, perfect vaccines, and use them as tools to deliver drugs and gene therapies. What are viruses? Viruses consist of genetic material, like DNA or RNA, wrapped in a protective parcel. Unlike most living things, viruses are not able to multiply on their own, so they need access to a living organism's cells in order to multiply and be harmful. When a virus infects you, it enters your cell, which mistakes the virus's genetic material for its own. Using that genetic material, your cell constructs the protective parcel and makes more copies of the virus's genes. The newly made copies of the virus can then go on to infect other cells and other people. What can we do with recombinant virology? Scientists begin by building genetic sequences that act as blueprints for the cell to construct viruses. These genetic sequences can get into cells using a range of techniques in the safety of a laboratory. The cells can then do the rest of the work, building entire viruses from the blueprints. While the viral life cycle may appear simple, multiple viral genes are often needed to carry out important functions, such as efficiently getting into cells, evading the body's natural defenses, or speeding up the virus's replication. Scientists can use a range of techniques to learn how different viral genes function. They can knock out and inactivate genes, which can reveal the function of that gene and whether that gene has a central role in the virus's success. Additionally, scientists can insert genes to use viruses as research tools, for example, by adding fluorescent genes to make the virus visible under the microscope. This can help us better visualize when the drugs we develop are doing their job at suppressing the virus. All of these different techniques can be used in a variety of ways to help us understand what genes make a virus dangerous or harmless, and design new therapies and vaccines to target those viruses. For example, if they identify features which are essential to the virus, then these targets may become the focus of novel therapies. Alternatively, if scientists remove a gene and discover that the virus is still fully functional, they know that we should not invest a lot of money and effort into drugs that target that gene. But if scientists are altering the genes of viruses, does that mean it's possible for them to create new viruses that could make us sick and potentially cause a pandemic? There are strict regulations and containment practices that researchers need to follow when they do recombinant virology. In 2021, an improved oral polio vaccine was made available to the public. It was created through years of accumulated research using recombinant virology and other scientific techniques. To learn more about the polio vaccine and how we uphold safety, see our next video.